Hello everyone, and this is our video on analyzing our microscopy pictures. There is another video on analyzing plates, slants, broths, and the like, and how to go about analyzing those pictures. This is not that. This is for Lab 1, I believe in both 2420 and 2421. The goal of this is to show you what you'll be seeing on your results PDFs, and then how to go about answering the questions associated with the exercises that we have you doing. So the first thing I want you to take a look at is, this is our drop page. This is the very first thing, and this has the title, exactly what it is we're looking at, as well as what our observations are done, what ones are under oil immersion. Remember, all 100 times objective lenses are done under oil immersion. The magnification is in the title of each slide. Each section slide will have the species. The stain we used for this is methylene blue. And keep in mind that if your worksheet has a micrometer, draw the picture with the micrometer. This is because when we're looking through our microscope, and the assumption is you've already watched the microscopy video, so you know what I'm about to talk about here. When we look through our microscope and we're taking pictures through the ocular micrometer, that itself is going to be of a higher magnification than the pictures taken with the camera in the microscope, because the camera in the microscope only takes a look through the nose piece objective rather than the ocular objective. So our first one we're going to look at is Cocoria rhizophilia, and this is a 40 times view of our stain. The majority of these pictures are mostly for you to see and show you what you're supposed to be looking at, and if you were to come into the lab, what it is exactly you're looking at each power. You don't necessarily need them for your worksheet. It's additional information for you to look at. This is 100 times. As you can see, individual cells are here. It's these small little circles. And this is the view through the ocular objective. And we have individual cells here. The micrometer allows you to measure. On some of your worksheets, not for this week, but certainly for next week and the following weeks going forward, you will need to provide measurements. If we don't provide a micrometer, you can't measure. So only measure on things that you have the micrometer. Almost all of your pictures that you're requested to draw are going to be using a micrometer because you want to measure on every single picture. Most of the pictures you're going to get this semester are going to look like this, where they are through the ocular objective with a cell phone. Here's Bacillus Magnetarium. As you can see, same thing, short rods. This is at 40 times. Here's 100 times. It's easier. And then this is the 1,000 times. Again, 100 times objective, 10 times ocular objective with oil immersion. Now let's take a look at this here. This is your laboratory guides and worksheets. And we're going to take a look at your first there we go. So this, I don't care what you draw. It doesn't matter to us whether you draw the Cocoria or whether you draw the Bacillus. That doesn't matter. What matters is that you draw the one with the micrometer and you answer the questions. Now, in the future, we'll tell you what you need. In the case of the majors, for the majority of your semester, it's going to be your unknown, and we will give you PDFs with your unknown. The PDFs can be quite large. The specialized staining PDF, you're looking at close to 100 slides. So you're looking at around 115 or so pages. It's up to you to find the right one. But when we take these pictures, it's going to have the micrometers, and always answer the question here. Cell shape and magnification you should be able to calculate. When we do calibration, you will want to include the total cell size as well. That will be necessary later on in the semester. For 2420, as I pull up your worksheet here, yours is going to be something very similar. Once again, here's your area that you're drawing. We want the name of the bacteria, cell size, arrangement, and magnification. I don't care which one you draw. It doesn't matter to me if you draw the Bacillus or you draw the Cocuria. It doesn't matter. What matters to us when we're looking at these is what type of drawing are you doing? Drawings in this case are not to be done digitally. We cannot prove that you didn't cheat. It's very easy to draw something on with an eye pencil here and then copy, paste it, and send it to everyone else. So it does have to be hand drawn. When you're answering these questions, you want to answer them to the best of your ability. If you can't answer something or we didn't give you the ability to, let us know and we can come to an agreement. In the future, especially when we start getting into things like how to look at certain, it'll be easier to show you actually on 2420, how to look at plates and growth characteristics, which is going to be lab 2, it's going to be difficult for you to answer some of these, particularly things like optical properties, which we can't do outside of a lab. If it's something that cannot be done outside of a lab, we tell you. We're not going to dock points because you can't get into the lab. We highly recommend you come into the lab and you see the optical properties themselves, or in the case of these, you look at the growth. Because the growth of some of these, I think I'm going to use, I'll probably use 3 chromogenic bacteria, or if I don't use 3 chromogenic, it's going to be 3 different bacteria. But let's take something like Cocuria rosea, which is our go-to chromogenic bacteria. It's a pink color, so we'd want that in the color. You won't be able to actually tell the optical properties or the size, shape, margin very well on a plate unless you can hold the plate and look at the plate. Because of this, we're not going to be docking points for you for things like that. So if it looks like there's a lot of data that you need and you can't get it, report what you see, and we'll go from there. The TAs and myself all have access to the pictures. And if we know that you can't see something, we're not going to dock you points for it. That's just cruel. And in this odd semester where we have to do this online when we normally wouldn't, that is a cruelty that we are not going to do. So there's no need to panic about the results. If you're in majors, there's no need to panic when you get a 115-page document. It just takes a bit more time. As a reminder on how the layout of our results documents, all the result documents are going to have a title and then all the information you need on the drop page. If there's more information that you need that can't be fit on the drop page, it will have a go-to on the drop page itself.